to counterbalance the injury caused. That's the properties of some microbes that again act as a balancing act between Rama and Ravana, which I talked about earlier. And here I would like to precise that the molecular genetics of nitrogen fixation and its regulation, on which I will talk in detail, is only a part in the vicinity of useful microbial diversity in nature that has been a recurrently growing area of proliferating research in health, hygiene and nutrition, physiology, biochemistry and biophysics, food science, toxicology and environment, agriculture, genetic engineering and biotechnology, cosmetic technology, fermentation technology, and allied industrial sectors. And rather what not, as the biggest tool to the interdisciplinary life sciences, to us offering protection, maintenance, and improvement to our quality of life. Or in other words, towards elevating the quality of plant, animal, human life. That's a kind of complete stewardship, coupled with this very, very systematic, innovative, meticulous and meaningful utilization by the scientific and technical experts, producers and consumers, with the ultimate aim to mitigate poverty by ensuring food and nutrition security. That's certainly a subject of paramount importance. To keep us at par with the rapidly advancing world of knowledge jewels in the national global scenario on the subject, its perspectives and strategies, overarching the goals of harnessing the unique properties of some useful microbes with a higher spatial resolution within the live green energy application sector, blended with a soft touch of fundamental science to trigger exceptionally well-planned, sophisticated researches to render commendable services to science and community, art and creativity, mankind and humanity, the grassroots level society, and the predominant knowledge-based national economy at large. And possibly that's the only means to learn the door of a scholastic paradise opening on this earth planet, the door of this earth planet. Rather, we should have the spontaneity and homogeneity to pick up all excellent components from every corner of the nation and the world at large on everyday basis in order to merge the fragrance and elixir of this with our own gem treasure key components to emerge, sustain, and to recurrently prosper and flourish like a world-class university wherever we are. And let me change. Yeah, the vastness of the subject can be just imagined such that even if we leave aside many other microorganisms and talk about or confine only upon the recently emerging concept of the mixed consortium of agriculturally useful microbes, or precisely the PGPR, that's the plant growth promoting rhizobacteria. It does include all the natural fixers, most of the phosphate solubilizers, most of the growth hormone producers, cedrophore producers, and most of the biocontrol chemical producers. And by further narrowing down to natural fixers only, we still find a wide array of microbial diversity, displaying a large number of diverse, associative, free-living microorganisms catalyzing nitrogen fixation for the entire biosphere, even in the symbiotic forms. And you know, catalyzing nitrogen fixation, that's an act by virtue of the two biochemical components of the highly oxygenic enzyme nitrogenase. And the enzyme nitrogenase has been highly conserved by dint of the evolution and continuity of NIF genes that codes for the structure and function of the two different components of nitrogenase. Evidence for certain nitrogenase-bearing bacteria present in present-day hot spring water and when Japanese volcano above 100 degrees Celsius, that shows that NIF genes might have been present right from the time of the origin of the Earth in the Precambrian era. There may be as many as 21 different NIF genes spread under 8 to 9 transcription units or operons, and the molecular cloning and sequencing of one of the 21 different NIF genes, that's NIF-H, I will show in detail, has provided a large, rapidly expanding database of sequences from diverse aquatic and terrestrial habitats. The biochemical and molecular genetic components of the unique enzyme nitrogenase and the highly conserved NIF-H gene, despite prominent diversity of natural fixes in nature, will be the focal theme of the deliberation. It is display little variations in the physiology, biochemistry, and genetic relation of diverse dasotrophs growing in nature and controlled under several conditions. Now see the paradox of nature. In the atmosphere, we have only 0.03% of carbon dioxide, and the entire carbon dioxide is consumed by the plants, all green plants, because of the asset they have, the ribosco gene, the photosynthesizing genes. But none of the plants have the NIF genes, that's the nitrogen fixing genes. Only certain bacteria and cyanobacteria are ornamented with this, they possess this. So this genetically determined discriminate use of CO2 and N2, because N2 gas is 78% in the atmosphere. The plants are not able to consume it because plants can consume only nitrogen in ammonia, urea form, some compound form, not in gas form, because it has a very, very strong triple bond between two N atoms. So there is only possibility to have genetic engineering in most of our cereals, pulses, pulses, vegetables, fruits, and flowers. That's the only answer at this juncture. Or we can make best use of our natural fixes, those bacteria or cyanobacteria which possess the NIF genes. Now you can see that if you use the synthetic fertilizers, these synthetic fertilizers are manufactured in factories at a huge temperature of 450 degrees Celsius. That's only possible to break the strong triple bond between two N atoms. And it is a Haber-Bosch process you must have read in your chemistry classes, right in matriculation. So that way we are spending a lot of very, very limited fossil fuel reserve. And the prices of nitrogen fertilizers are therefore escalating very, very high. And the second truth is that 75% of synthetic fertilizers, apart from the cost that comes in transport of the fertilizer from factories to the fields, this 75% is consumed in the vegetative growth of the plants. I mean, stem, leaves, etc. And only 25% goes from source to the sink, grain production. And what happens that 103 elements are required for plant growth, and we supply only nitrogen plus phosphorus and potash. Some progressive farmers give zinc and boron, etc. But what about other elements? These are in the same proportion is squeezed out of the soil, and soil becomes sterile day by day. That's another big problem. On the other hand, if you use those organisms which bear the NIF genes, I mean the biological nitrogen fertilizers, nitrogen fixers, they require nothing but only solar energy and water for the multiplication. They have their own ATP generation system, reductant supply to convert elemental nitrogen, that's atmospheric nitrogen, into ammonia. This is very well metabolizable by the plants. And these are self-renewable, most economic for biotechnological production, distribution, and use. And another beneficial point is that it has improved through radioisotopic studies that 90% of the bioproduced nitrogen fertilizer is diverted to the sink, for the grain production. So plant may be smaller, but the quality of food grain will be very, very high. And it also produces the growth hormones to help restoring and maintaining, enhancing the soil fertility. To see the difference, the only thing is that we have to work in coordination with the agronomists and microbiologists. It's not only that we have to straightway apply the agronomic recommendation of NPK and other fertilizers without testing the soil microflora, how many nitrogen fixing organisms are there, NIF bearing organisms are there. Now, if we have a rough estimate, the industrial fixation which we do through Haber-Bosch process is a maximum 60 million tons every year, for which we invest a lot of fossil fuel for creating 450 degrees Celsius of temperature. Through lightning, we have 5 million tons. Through human and animal waste, we have very small, and green mirroring 15 million tons, but it's only the biological nitrogen fixation, whereby we have 180 million tons of nitrogen produced in the form of ammonia and urea. But these are scattered. We have to make best use of it in coordination with agronomists, soil scientists, and microbiologists. Now, the specialty of nitrogen fixation, the time is very short.
Yes.
Sandeep has given only 40 minutes.
Yes.
So I should say that this gene is available only in certain prokaryotes, some bacteria and cyanobacteria only, not in any eukaryotes, higher plants or animals. And most important is that it is highly inoxygenic process, means it is oxyphobic. It doesn't work if the threshold level of intercellular oxygen rises above a considerable level. Otherwise, it destroys irreversibly the nitrogenase enzyme and the NIF gene. And the last one is that it works only under a nitrogen crisis. If there's no ammonia or little ammonia, only then the NIF gene will be triggered to start coding for nitrogenase enzyme to form ammonia. Now there are free-living nitrogen fixers like strict anaerobes. These are those anaerobes seen today in hot springs of Rajgir, Kullu, Manali, even in volcanoes of Japan and other places at very high temperature. And the NIF gene is alive, working, that gives an index that probably at the time of origin of the Earth, this gene was present. It's miraculous to think. And the Methallococcus, Metallosarcina, these are the two bacteria and cyanobacteria, which are present today, and they reflect the old history of the gene. And eubacteria means the same character in the present bacteria, present at ambient temperature. Now, facultative anaerobes are those anaerobes which are otherwise aerobic, produce oxygen also in their life work. But whenever there is nitrogen crisis, they adopt to anaerobic mode, like Klebsiella pneumoniae and other species. And Klebsiella is a model bacterium so far as NIF genetics is concerned. How this works, I will tell you in the coming slides. Now, there are the microbes which are producing only little of oxygen. So that oxygen is not preventing the NIF gene activity. So these are known as microbes of considerable origin. Then we have pure aerobes. Pure aerobes mean oxygen is being produced. There's a mechanism going side by side how the NIF gene is protected in working form. I will tell you later. Pure aerobes with non-oxygen evolving system, that's no problem. Rhodopseudomonas species are there, Rhodospirillum is there, so there's no problem, no oxygen evolution if it is secured. And pure aerobes, BG and cyanobacteria, these are having oxygenic photosynthesis just like hard plants. It's interesting to see how this gene is protected here. Now, associative nitrogen fixers like Azospirillum, Pseudomonas, they fix nitrogen in the close proximity of the roots of plants. It will be interesting to see how the mechanism of oxygen protection is working there. Now we have symbiotic groups, various groups of rhizobium known as cynorhizobium, that's a redesignation that infects only the pulses. The bradyrhizobium also infects the pulses, but it's slow growing. And the privilege is that it can fix nitrogen even in the test tube in vitro condition. And that facilitates deciphering the science of NIF gene inside the laboratory. Then we have other species in lotus and other non-legumes. And last one is azorhizobium. That's very interesting. It doesn't only fix nitrogen in the root nodules, but also in the stem nodules. The only host is Sesbania rostrata. Then this is another interesting slide. How there's a matching between host and parasite. The plant root hairs produce lectins, and the rhizobia produce ricadhesin. Unless these two match, the infection cannot be there for NIF gene to be transferred and view to be formed. And same thing is with COVID-19. It searches out its exact human partner. That's why there's a lockdown. If you move out, the corona will mark you as the exact match and infect you. If you locked yourself inside the house, it cannot mark. It is just like ladies purchasing red sari from a market and moving from shop to shop in search of a very matching blouse piece of the same color, while there are hundreds of red colors in the market. Unless you get a particular match, you cannot accept it. The same way corona have many animals in the environment, but it searches out for the human being. So please protect yourself. This is the only message for this slide. Now, these are the symbiotic forms which are feeding nitrogen in non-legumes like Frankia. The not such bacteria are called the Frankia species, Elder, Casuarina. They are also doing the same work of nitrogen fixation. Now, Cyanobacterium is the only organism which has symbiosis with almost all categories, right from fungi in the form of lichens to bryophytes, pteridophytes, gymnosperms, angiosperms. Pteridophyte azolla is the best example on which our lab has worked, produced many mutants, tested far and wide for its thermo-insensitivity, low phosphate requirement, and spore production, so that you throw a handful of spores in waterlogged rice fields and to develop into plenty of azolla, and you don't need any nitrogen fertilizer from outside. Now, about oxygen protection mechanism, you should see that in Cyanobacterium there is a heterocyst. Whenever there is nitrogen crisis, a het gene is triggered and it forms a heterocyst as you see in the middle. And here, the photosystem second is absent, where oxygen is involved. Only photosystem 1 is present, whereas through cyclic phosphorylation, ATP is generated. Reductant is supplied from the nearby vegetative cells. So it's a kind of symbiotic association. Lot of nitrogen is fixed inside the heterocyst and supplied to the nearby vegetative cells. And so far, people were of the knowledge that non-heterocystous cyanobacteria are non-nitrogen fixing, but it's not so. Latest discoveries have shown that NIF gene is present in every cell of non-heterocystous bacteria, that's cyanobacteria.
Sir, sorry for interruption. Actually, we have running out of time, so please, we have only 5 minutes. Yeah,
I'm summing up. Yeah. So these are the oxygen production mechanism, that's the auto mechanism going on inside. Now, coming to the NIF gene, you see it's a 24 genes present, that's known as one NIF gene. It has 24 components. The importance is its regulatory factor, that's NIF A and NIF L. NIF L is the negative regulation, that is showing the sensitivity to oxygen. Whenever intercellular level oxygen is high, it will be switched off. And NIF A responds to the amount of ammonia present in exterior. If amount of ammonia is less, then it will trigger all the genes to switch on. So this is the whole genetics of nitrogen fixation to understand clearly. And this is the continuous unit, the ZIS A and ZIF units in Cyanobacterium, which makes the whole NIF gene into a continuous unit to make NIF gene to work no sooner there's a nitrogen crisis signal received. And there's the same gene pattern in the symbiotic system. There's also in symbiotic system. There's a shortcut. If there's escape of nitrogen and hydrogen, it will be returned back to the system. This way you can produce oxygen resistant or ammonia resistant. There's all this nice work has been done during the last decades. And NIF gene genetic engineering, what I told in the beginning, this effort is also getting successful day by day. From this organism to the other cyanobacterium, then to the bacteria and yeast, we have touched the eukaryotes, and then we have come from eukaryotes to the plants in the chloroplast, because chloroplast DNA is comparable to the whole cyanobacterial cell, which has free DNA, free thylakoids, so, such as the chloroplast, which has free DNA, free chloroplast. But here, the oxygen is the only problem, and these are the various organisms we have been trying. And I will show, this is the cloning and sequencing of other nitrogen structure genes. How we can make NIF-L insensitive to oxygen so that nitrogen can be produced, fixed, even in the presence of excess of nitrogen. And how superoxide dismutase enzyme can be formed to take care of excess oxygen, and how we can transfer NIF gene to the mitochondria type organelle where there is no oxygen evolution. So we are here at this stage, and maybe that in the days and years to come, we are able to transfer the NIF genes to the plants itself to make them independent of nitrogen fertilizers. Thank you very much. Questions, if any?
Okay, thank you, Professor Vaishampayan. So quickly, if anybody have any question, then please, one by one. So I'm going to unmute all. So please ask a question one by one, if anybody has. Any question? Hello. So if yes, so if anybody have any question, then Professor Vaishampayan is here. You can ask any question if anyone have any doubt.
You can give them a mail ID also if later on.
Yeah, please. So please note down the email ID, Professor. vice.chairman.iqac.bhu@gmail.com. So,
can you see the screenshot?
Yeah, I think everybody can see the screenshot. So if anybody have any question, he or she can write to Professor directly, and one can solve their question or query, whatever they have. So I think that
repeat the email ID.
You cannot see the screenshot of that PowerPoint.
Okay, sir.
Okay, note it down. That is vice, VICE .chairman.iqac.bhu@gmail.com. Should I repeat again?
No, sir. It's good. Thank you, sir.
Okay. So if anybody have any question, then just ask very quickly because we are running out of time. So if anybody have any question, then please.
Dr. Sahab, I want to ask a question.
Sure, welcome.
Yeah, sure.
Myself, Dr. Sarvesh, Director NTSP at ARA. I am very much impressed by this very fruitful lecture of Dr. Vaishampayan, sir. And I came to know that ladies, how they are very much adamant to settle their costumes according to the needs. That is why the corona presses the bodies of its matching. Actually, I would like to ask, how could we improve our immunity while going inside our room by preventing ourselves under the Lakshman Rekha nowadays in lockdown system, so that
immunity improve करने का तरीका कुछ है?
we could, how could we improve ourselves, our immunity, so that we can cope this type of virus, because there is no medicine of virus nowadays. Sir.
Good question, inquisitive question. You are not going to improve your immunity against corona, but you are going to protect and save your immunity, whatever you have, whatever God has given to you and whatever you have maintained through yoga and through your food habits and other things. So you are making it intact so that if at all you are exposed to corona, it may help you to combat it as far as practicable,
Yeah.
until and unless the vaccine comes.
Okay, thank you.
Better to protect yourself and guide your children and aliens in the house above 60, 65 to retain themselves inside the house so that your immunity is intact. And if at all there is an accidental chance that you're exposed to that, it will takes its own. So that's all. That is what I can say.
Okay,
Any-
sir. Thank you very much.
Okay, thank you. Thank you, Dr. Sarvesh. If anybody have any other question? Yes, please, Swarna. Yes, please.
Yes, actually, yeah, so I'm speaking from Bordeaux University of France, and I have a question to Dr. Vaishampayan. I was listening to a very nice talk given by him, and I have always been listening to his lectures. So I wanted to ask a question regarding the nitrogen fixing bacteria's NIF gene that is in, he told, he was talking about Azola. So I just wanted to ask about the genetic plasticity of this NIF gene, whether these genomes have whether he has explored for other nitrogen fixing cyanobacteria like Nostoc cycad present in the colloid roots of cycas and other things, and can these also be treated genetically like he has done experiments, he is doing experiments with Azola.
See, good question. The NIF gene in cyanobacteria, as you know, you have, I think you have been in cyanobacteriology. Now you are in France.
Yeah.
I know you very well. So you know the NIF genes in cyanobacteria is scattered. It's not a continuous unit, as you find in Klebsiella pneumoniae. So the study on NIF genetics in cyanobacteria is only from academic point of view, and from the important point of view that how we can protect our NIF genes from the oxygenic photosynthetic system. Otherwise, so far as gene transfer is concerned, it's always done through Klebsiella pneumoniae, where you have all 23 units of NIF genes together. And all can be transferred as has been done to E. coli. We have in the library of University of Maryland, our genetically engineered Escherichia coli, which is otherwise non-natural fixing, but now a natural fixing E. coli is present in Rockville, American Type Culture Collection Center. And everybody takes it from there for further research. In cyanobacteria, what you say, the genes are already stored. The NIF genes are stored, but it's not for transferring point of view, transfer to higher plants, because it's not a continuous unit. Unless you add the ZIS-A and ZIS-F recombinase genes to out the unnecessary genes in between and make it a continuous unit, you can't make it functional even. So to make it functional, we use the recombinase genes, but for transfer of genes, it is very difficult from cyanobacteria. The only possibility is Klebsiella, because in Escherichia coli, you do not have matching sites where the NIF gene can be attached. In yeast, there was no matching site. Yeast walking plasmid we have transferred. But we have befooled the yeast system by first transferring the E. coli plasmid inside the yeast system. So that it thought that I'm having E. coli gene. It has a recipient system. And in between the E. coli genome, we put the NIF gene from Klebsiella. And that's why a nitrogen fixing yeast could be discovered. But it's such a small system that a huge iron and molybdenum requirement of nitrogen fixation cannot be met by the yeast. Otherwise, it will jeopardize its own system. That's why we have gone a step ahead to direct microprojectile bombardment in the higher plants, the fluoroplast system. And now people are trying the mitochondrial system.
Okay,
Thank you
now,
very much, sir. Thank you very much.
okay, thank you, Suvarna. Now, next question is Dr. Anil Kumar Singh want to ask. Please.
Yeah. It was an excellent presentation, and we learned a lot about this nitrogen fixation bacteria and microbes. Despite having this much potential, why this nitrogen fixing bacteria and other biofertilizers are not popular in farmers community? Means they are not having adoption as much as it should be actually. So what are the reasons why it is not happening?
Yeah, I told in the very beginning, in India particularly, if you go to the western side, in US I've seen fields almost every year I visit, you have the crops standing of all equal size and equal look, because they make use of biofertilizer very meticulously. Here the problem is that we do not have a requisite coordination between agronomists, microbiologists and soil scientists. And straight away we apply huge quantity of nitrogen, especially taking care of the high yielding varieties. With the result that, as I said, NIF gene is triggered only when there is a crisis of nitrogen. If you put a lot of synthetic nitrogen, it will in itself will switch off the NIF gene, NIF genetic mechanism of the bacteria. So there is a paradox, great contradiction within ourselves. So our farmers are gradually being trained, and our scientists also need to be trained. At least from the younger generation, we have high hopes that they will have the interdisciplinary, transdisciplinary, and multidisciplinary researches. Unless we adopt all these, we cannot get through, although some progress has been made, but little only till date, you are right. But we are trying hard. ICAR has made several branches of agriculture researches, nitrogen fixation researches. Krishi Vigyan Kendra, hundreds and thousands are there who are going to apply, receive the authentic biofertilizer pockets from the IRI and other centers, and training the farmers how to use it. So a day will come, it will say, the biofertilizer will see the light of the day.
Is it clear now, Dr. Anil?
Yeah.
Okay, thank you. Thank you. So now next question, the Supriya Singh want to ask. So please, whatever question you have, you can ask. And then after, Dr. Ashish. Hello, Supriya Singh is there? Okay, Dr.
Yes, sir.
Okay, then please ask the question.
So can we use membrane recognition site for the development of vaccines?
Yeah, that is what's being done by a few companies. At least from animal sources, they are receiving because everything is genetically controlled.
क्वेश्चन कर रही है अभी.
Unless you have the permeability, necessary permeability required for the vaccine to work outside the system and inside the human system, you can't work. All membranes cannot work. There is a varying proportion of positive and negative charge on each membrane. So these are all under trials, whatever you have asked. Many sectors are trying.
Okay. Thank you. Now, Dr. Ashish, please.
Thank you, sir. Such a nice presentation. I am Dr. Ashish Kumar from Western Australia. I have a very common question. So, can we fulfill the nitrogen requirement just using green manure? I'm asking this question with farmers' point of view. So, can we avoid the use of synthetic nitrogen? Can we just use the green manure to fulfill the nitrogen soils nitrogen requirement? And if not, then how we can improve the nitrogen content in green manure? Yeah.
That's a very good and pertinent question.
Yes.
You know, I've shown green manuring and human animal waste also. Through green manuring, you can replenish your soil. That is the one positive aspect. Whether you can completely replace the use of synthetic fertilizer or not, we cannot say at the moment. Although we have in Sikkim, Uttarakhand, or throughout, all organic fertilizer-based crops are growing, as they claim. And to a great extent, it's true also. But, you know, India is still a not so well-to-do country. Here the problem is how to grow more and more to feed the exploding population. You are 135 crores, rather more right now, and population growing every day. So to have a greater quantity of food produced, we have to use the high-yielding varieties of wheat, rices, pulses, wild seeds, fruits, sprouts, vegetables, every sector. And high-yielding crop varieties, whenever you use, they require a very high quantity of fertilizers, especially nitrogen. And that high quantity of nitrogen cannot be made only with biofertilizer. It can be supplemented, provided you take care of the amount of synthetic fertilizer being used, which is not suppressing the NIF gene presently. Otherwise, the nitrogen fixer itself will convert into a nitrogen consumer because the NIF gene is so that it is self-regulated. No sooner a nitrogen level, fixed nitrogen level is higher in the environment, it will switch off. So a lot of scope of science is there. Now, from quantity to quality, we have moved. We have shifted our research to quality improvement. And biofertilizers are the only means to improve the quality or any crop, like even pesticide-free, herbicide, fungicide-free, because the cyanobacteria and bacteria we have now, which produce biocontrol agents, means natural fungicides, insecticides, herbicides. So by a proper coordination between these biofertilizers and the synthetic fertilizers, you can shift from quantity to quality. But presently, to feed the exploding population is a great problem. You can understand the present scenario is of the nation, not if not of the world at large.
Okay, thank you.
Thank you, sir.
Thank you, Professor Vaishampayan. So I think now you can switch off this screen and you can come on the camera so that everybody can see you, and then he or she can ask a question. If it is possible, sir.
But how to come on camera? That's the problem.
Just click on share screen. It will go.
Okay, let me see.
Yes, now we can see everybody on the one panel, and waiting for Professor Vaishampayan to come on the screen. And if anybody have any other question, then he or she can ask.
Sandeep sir, my son knows Vaishampayan sir, so he wants to say a hi to him.
Okay,
so
okay, that's why. Yeah, that's what I'm requesting Professor Vaishampayan that please. Yeah. So he's trying to come on the camera. There's some problem. Yeah. So I would like to tell something about Professor Vaishampayan. He is also an advisory board member of my foundation, that is Indian Scientific Education and Technology Foundation. And Dr. Ashish and Dr. Anil, he is a life member of the ISET Foundation. So, and basically this foundation is working in the field of science and technology promotion and in the rural area as well as suburban area. So you all people are belongs to the scientific community. So I'd like to request you all that please come forward and join this foundation so that we can do a lot of things in the rural part of India, because the science which is very much needed to promote in the rural part of India. We have a great minds in the rural part of India, but they don't actually have some guidance or some lack of promotion, scientific promotion or motivation. So that's what our foundation is trying to promote, the scientific education and the quality of the economic agricultural things for farmers, how they can do the scientifically their agricultural, how can they do the economic things. So in this field we are just doing, and Professor Vaishampayan always motivating me and always helping me in all the way. So I'm really thankful for giving me opportunity to provide such a great panelist like all over the world. I think somebody is from France, somebody is from Australia, somebody is from India, and I really love to host this seminar, and I always thankful to Professor Vaishampayan. So and I always thanks to Dr. Assis and Dr. Anil too for motivating and helping me to plan the things. Recently, I have some organized, drawing competition for the students, and we have received more than 150 drawing from all over India, and just now scrutinizing them, and just on the 7th, I will display the result. I have opted some prize for that competition, and we selected the 3 drawing, and we awarded with the certificate, with the certificate and some prize money. So I also thankful to all the participants who have given their important time, and I can see all of you on the screen. So if anybody still have any question, then Professor Vaishampayan is still with us. You can ask. Even you cannot see them because I think there's some problem in the screen. So
I have a question.
Yes, please.
Is there any vaccine for COVID-19?
Oh, good question, Professor Vaishampayan, please.
Yeah, I couldn't hear.
You cannot hear?
Yeah, no, the voice was not audible. You can just repeat it.
He is the son of Suvarna, and he is asking that is there any vaccine for COVID-19?
Yeah, there are 2-3 vaccines on way. These are under trial, and please apprise him of the fact that unless it is properly tested in human beings, so that it doesn't have any after effect, any deleterious effect, only then it will be allowed to be commercialized, to be given to some pharma company to produce to the people. So yeah, of course, vaccines are on way.
Thank you. Any other question?
I have one question.
Okay, please, Dr. Soumya. Yeah, please.
Yeah. First of all, very good morning to all of you. Really get no, for the first time we say good morning. So it's a good morning from my side.
Yeah.
Because most of you are new to me. So one thing I would like to ask. First, I would like to thank Vaishampayan sir for giving me this opportunity to get the knowledge of this slide. And of course, one thing I would like to ask about COVID-19, there is a lot of news on Facebook and social media that the COVID-19 is mutating and it is changing its symptoms among humans. Is it true? Or many of American patients who were suffering from COVID-19, their kidneys have failed during the course of medication. Is it true or it is all fake? I'm not very sure.
Yes, very good question.
Good question, yes.
You see, the type of changes in corona is not so much as we are still unaware of the total science. People so far think that it attacks the lung system only, blocks the lungs. But from the beginning, it has a deleterious and injurious effect, not only on kidney, on your heart, on your blood vessels, etc. That's why you have to save everything, because there's a receptor side. Everywhere you have a receptor, ACE2. That receptor, if it is in a larger quantity, it will receive like a ball and socket joint. It will pick up the corona cell, and it won't allow it to move to the vital organs like heart, kidney, liver, and lungs. But if you lose your immunity and move fast, move here and there, then you will lose the number of ACE2 components. And therefore, corona may be harmful. Now coming to the various strains of corona, of course, you know it's a virus and it's inherent nature of the virus to recombine with the genome of the host it attacks. So inside the human genome, it recombines with the human DNA. In itself, it is RNA, but it recombines with a portion of DNA anywhere, random, and it will leave its own genetic component, and in return, it will receive the same number of nucleotides from the human DNA. And then it is a changed virus. You can, of course, say it as a mutant, but that is not so harmful. There are only four types of corona: alpha, beta, gamma, and delta, of which alpha has been found in the tigress of New York recent. It was a big news. Also in some cats. Beta coronavirus is the only strain known so far which is injurious to human beings. And rest of the variants are simple. It is just because of recombination with the human DNA. So human DNA cannot harm the human DNA. So rest assured. But of course, you have to save yourself from beta coronavirus. It searches out, as I said, sari and blouse piece match. It searches out the human beings only. Alpha coronavirus is not so injurious. It may be in mammals, animals, but mammals. And delta, gamma, etc. are with the birds. You had a few years back swine flu, etc. They were due to gamma and delta coronaviruses. I think that's a sufficient answer because I know you are a student of science. I can understand much.
Thank you.
So I think this is the time to say thank you, Professor Vaishampayan. Is there any question from anybody? Please make it quick because it's already a 1 hour call. If anybody have any,
I have a small question. Yes, I am Swarna Kanchan. So I have gone through a number of literatures. People are working on COVID-19 vaccine development. Still, people are using traditional method of vaccine development. People are not using computational methods, which can intensify the vaccine development process. People are not using why it is happening.
Yeah, good question. Because you know there is urgency. Unless we pass through the time-tested methods, how to leave the time-tested methods, which always has a better chance to yield sooner. And why to shift to the computational method right now? They're a group which are trying the computational method. It is still to be standardized. It has not yet been standardized, so much so directly used for the production of coronavirus vaccine. So that's why traditional methods, which are time tested, are being used and little success has been made.
Thank you very much, sir.
Thank you, sir.
okay. Now I must say, yes. Yes, anybody have any question? Please. Hello. So I think no one has any question. So I really thank to Professor Vaishampayan for his valuable time, and I am also equally thankful to all the participants who have just attended this webinar, and we'll come soon with another webinar session from the professor from the USA who is working on neurobiology. So we'll probably host by 10th May or 15th, in between 10th and 15th May. So I will post on the WhatsApp group and Facebook group. If anybody wants to join, then most welcome. So now with this words, I would like to just end our session, and I will really thanks to all. And I am very much thankful to Professor Vaishampayan. Thank you so much for your time, sir. And
thank you, Dr. Sandeep.
Okay,
thank you very much.
So bye to all of you. See you soon in next session. Okay, bye.
Thank you, sir.
Thank you, sir.
Thank you, sir.